Over the course of the last few years, COVID-19 has popularized a seemingly obscure scientific term called PCR. It's become a household buzzword of sorts. Polymerase chain reaction, or PCR for short, became a phrase everyone started tossing around. And while people were visiting clinics to get a PCR test done to find out their COVID-19 status, laboratories across the globe were busy running those tests and pumping out results. PCR, put simply, is a laboratory technique used to amplify and analyze DNA. It's become the go-to method for detecting the genetic material of the coronavirus. Perhaps what people don't know is that PCR is so much more, and has been for a while. In this video, we'll be joined by an expert from the field who's going to help us learn more about PCR, how it's used, how it has been used, and what the actual mechanics of the test are. So, without further ado, let's take a trip through a day in an infectious diseases testing lab.
PCR stands for Polymerase Chain Reaction. So if you can imagine, if we have a complex specimen, we take a nasopharyngeal swab from the back of your throat, and it has all sorts of bacteria, viruses, human cells, but we want to know if there's, let's say, influenza there or COVID there. So the steps would be, first of all, we take a specimen, a clinical specimen. It could be blood, it could be cerebral spinal fluid for meningitis, encephalitis, it could be a nasopharyngeal swab for a respiratory infection. There are various things that we'll use for different clinical syndromes. So we take the specimen, it has to be transported in a way that the specimen remains intact, that the nucleic acids are preserved. And then in the laboratory, our first step would be to take a sample of that, and we would extract the nucleic acids, the DNA and the RNA. And by purifying them, we get a better reaction. We try to get rid of the protein and the fat and anything else that's there. We then would take a sample of that DNA and RNA. And if we're interested in RNA, we would first change RNA into DNA. That's an enzyme called reverse transcriptase. And so this reverse transcriptase will change your RNA into DNA. And then the DNA is a template for this reaction we call polymerase chain reaction, or PCR. In PCR, we have a couple of steps. First, we have to have designed primers that are specific for the infection of interest. So, for example, if it's COVID, we have primers for some part of the genome, and for that we need to know the genome. And knowing the sequence, can we design a PCR where there are conserved regions that a short primer can stick to. And so the process is, we've got this extracted, let's say, RNA, we change it into DNA, we take that DNA and we use heat to separate the two strands of DNA so that you've only got a single strand of DNA at a time. Next, we take our primers, we add it to the mix, and these primers are going to stick specifically bases they recognize, and they form a template where we're now going to fill in what's in between. And what's in between now gives us a product. And what we've now done essentially is cloned a small part of that DNA. Most reactions we might be starting with 10 copies or 100 or 1,000, and one cycle won't give us enough that we can detect it. So we're going to go through 30, 40, 45 cycles of this, where we split the DNA, we attach the primers, we elongate it, we get a product out. And after we've done this 20, 30, 40 times, we might have product that we can see. Polymerase is an enzyme, a protein that makes some sort of polymer. So in the setting of PCR, we have a DNA-dependent polymerase. It uses DNA as a template to make a further DNA strand. It's copying a single strand of DNA and making it a double-stranded DNA. So we mentioned that to be able to do a PCR test, you need an appropriate sample, but then you need to put it into a medium that keeps it stable. For PCR, we want the RNA and the DNA to not degrade over time. And so if you take a sample and you get it to the lab in an hour, it won't degrade. But if we're getting samples from other laboratories and perhaps we're backed up or they're backed up and sending them out, and it takes maybe 2 or 3 days to do the PCR, and that happened when we got very, very busy during COVID testing. If the medium allows degradation of your nucleic acid, you might not be able to detect it. So transport media are there to try to stabilize that, although we can also simply refrigerate things. So the McMaster Molecular Medium was something that only allows molecular testing by stabilizing the RNA and DNA. But for PCR testing, that gives a very, very robust, very reproducible result, whether you do it in 1 day or 1 month. So that was a big, big advantage. We were very concerned with COVID testing that the actual specimen could be infectious to our laboratory staff. MMM was invented in part to kill the virus, and we were able to show that within about 5 minutes of putting a virus into MMM, it could no longer be cultured. And so that would provide a level of biosafety for our laboratory staff. So this was designed by Dr. David Buehler here at St. Joe's, and it was developed in order to stabilize, in order to inactivate, and in order to make it a lot easier to do high throughput automation with the robotics that we use.
PCR is arguably an essential when it comes to infectious diseases testing in laboratories. Our hope is that you know more now about PCR than you did before watching this video, and that we were able to show you exactly why you were getting a stick stuck up your nose during the COVID-19 pandemic. The PCR test is constantly evolving with technology to be more efficient every time. And with it, our understanding of the viruses in our community evolves as well. A big thank you to everyone that was involved in making this video, from our expert who we interviewed to our lab technicians that were filmed.